Okay, I'm going to go over this. This is PowerPoint 10, Part 1, and they're talking about gene interactions. They're going to talk about dominance and recessive mutant, or null recessive mutants. Let me just turn my stuff off so no one messages me. Okay, so forward genetics is when you have a gene of interest, like a phenotype. So, for instance, these red, yellow, blue, whatever, these bell peppers, they all have a different color, and so you're trying to figure out what gene causes that phenotype. So what you do is you take that color and you try to mutate certain alleles that you think might be causing it. So you have a gene of interest, you have a phenotype of interest, and then you mutate the actual, you like select for certain things. So this is different from reverse genetics, where you are trying to figure out what that allele or what that gene is actually affecting. So you're going from the genetics to the phenotype. In forward genetics, you're actually going from the phenotype and you're working with the specific genes to try to produce that phenotype. I hope that makes sense. So dominance, how we are used to seeing it, is that all you need is one allele for that dominant trait to be shown. And there's several types of dominance, but complete dominance is what we're used to. And so here we show the complete dominance. So if you have a homozygous wild type, it's got two dominant alleles and it produces a chromosome. Both chromosomes will produce functional proteins, and then the whole thing will function. And this is, again, all of these are going to be displaying complete dominance. So here you get a mutant allele, but you still have that other dominant allele, so you produce a non-functional chromosome, but you also produce a chromosome that can produce a functioning protein. So this chromosome produces a non-functional protein. But because the functional protein is still in the genotype, you get a phenotypically functioning protein because the dominant allele is overpowering the mutant. And this is, again, homozygous recessive, which is what we're using. This is, again, complete dominance. You get two mutant alleles, and that means that you don't get a functioning protein. There's nothing to overcome one of the non-functionals like there was in this example. So you get a non-functioning phenotype. And that's just the normal dominant versus recessive. That's complete dominance. That's what we're used to seeing. And so that's only one copy is necessary for that phenotype to be present. And that's like, this is homozygous, this is phenotypically seen, but you also get this phenotype produces the same thing as that one. I think this is the most easy, or the easiest one to understand. I think probably we're all confident in understanding how complete dominance works. So mutant alleles can be fully dominant over wild type. So you can get mutant alleles that have dominance. Wild type alleles are often fully dominant to mutants. So yeah, the wild type alleles are often the ones that are fully dominant or that are phenotypically normal versus a mutant allele. But sometimes you can get mutant alleles that display that complete dominance as well. So in incomplete dominance, this is when you get that dosing effect. And I think it's confusing sometimes because incomplete dominance and co-expression seem similar, because co-expression or codominance is when both alleles are being seen, but this is different than incomplete dominance. Incomplete dominance is saying that you've got a scale between two phenotypes. So if you have purple flowers on one spectrum and you've got white flowers on the other spectrum, and you have a purple and say purple's dominant, and you have a white, which is recessive, if you have a purple and a white, you might get a pink. Where if you have two purples, you'll get a purple, and if you have two whites, you'll get a white. So they're saying that there is variation between pink or purple and white. And so you get an intermediate parent. each wild type alleles, gene products contribute to a dose of the phenotype. So we sort of talked about this in another lecture where we talked about how you could have several different variations of a specific genotype. So incomplete dominance, neither one is dominant over the other.
There you go. What are you doing? Turn up drinks? Thank you. You're welcome.
Complete dominance versus incomplete dominance. So here we see it again, and you get a phenotypic range in complete dominance, where you either get this red, you get a red flower or you get a white flower. There's no in between. You don't get a pink flower. That's complete dominance. If you have AA, I hate when they do it like this. Say this is big A, and this is little a, little a. If you get big A, little a, you get a red flower. And incomplete dominance, neither is dominant. So if you have A1 and A2, or if you have a heterozygote, you are going to get some variation of the gene. And it just depends on how those genes interact. Sometimes they could be controlled by more than one, but incomplete means neither one is dominant over the other one. Codominance, however, is when both alleles are going to be expressed. You see this in blood types, and you see this in sickle cell anemia. A person with sickle cell anemia, if they have they're possible to have both normal cells and the sickle cells. So these are a little bit different, sometimes maybe a little confusing, but if we move on to this, we can see it in blood types. So you get the codominance of antigen products. So your blood produces antigens. They can either produce antigens for A, or antigens for B, or antigens for both A and B. And they show you that down here. So genotype AA, AI, you're going to get blood type A. And blood type A means, so blood recipient reactions to donor blood. A produces antibodies for B. So if you have type A blood, you cannot take anything that has B blood because you don't have antibodies for the antigen B. Wait, that may be wrong. Hold on. A antibodies. Okay, yeah. Sorry. So let me see this again. Donor, phenotype, blood, A, genotype, AA or AI, antigen type A, antibodies made by body. Okay. So if you have type A blood, your body makes antibodies for B. So a person who is A, has A blood, has B antibodies, and B antibodies will kill off anything with B. So you have to give this person A blood or O blood. So let's look at another one. B has either double B or BI. They get antigen type B, and they make antibodies for type A. So they're going to kill off A. This is like antibodies kill, right? They protect your body against foreign cells, foreign objects, foreign whatever, foreign antigens. So if you have a donor with B blood, you can't give them type A blood. That's what this is saying. So A can get A, B cannot get A. AB can get both A or B because it has no antibodies for A and B. So it's not going to kill off the B cells in the AB blood. Donor type blood recipient reactions to donors. So red blood cells that do not react with the recipient antibody, that's these, remain evenly dispersed. Donor blood and recipient blood are compatible. Blood cells that react with recipient antibody clump together, and donor blood and recipient blood are not compatible. So you start to get clots when you mix things that aren't compatible. So in O blood, you have A and B antibodies. So your body if you have O blood, is killing both A and B. So you cannot give O-type blood. You can only give O-type blood to O-type people because it'll kill off anything that has A or B in it. And AB has both antigens. An antigen is a foreign material, and if your body has an antibody for it, it'll find that foreign material, or it'll find that antigen, attack it, and kill it. That's why you have to make sure you give the right blood to the right people. AB is a universal receiver, so that's this column here. They can accept blood from any donor because they have universal recipients. They don't produce any antibodies, so they can have any blood. No antibodies produced. All right, so let me go over that again because I think I made that more confusing than it needed to be. But if you have an A recipient, you're trying to figure out what blood to give them. They create A antigen, that they have A antigen in their blood, but they create antibodies against the B antigens. So you can't give them anything with B antigens. And B and AB both have B antigens. But the A recipient does not make antibodies for B blood. I'm sorry, it does make antibodies for B blood. So when it sees any kind of B in the donor blood, it'll attack it, whether or not it has A or not. It will attack anything that has B in it. Since O doesn't have antibodies for since O doesn't make antigens for either type of antigen, A or B, it's free of antigen, so it has no foreign material, and it can be given to anyone. That's why it's called the universal donor. Okay. B has A antibodies, which means it can only take B blood, because A antigens are going to attack anything that has antibodies are going to attack anything that has A antigen in it. I feel like I'm on a rant here. So I think this is the part where you need to study. You need to just remember that AB has no antibodies, so it's not going to kill any foreign blood. And O has both antibodies, so it's going to kill every blood type except for O, which has no antigens. So think about it like this is the foreign thing, or you could call it a virus. It's not a virus, but you can think about it that way. Or maybe a germ, call it a germ. And this is your antibiotic or your anti-germ. So this is going to kill germs. Kill germs. And I don't know, study this page. This is the page that's going to help you. The main concept I think here that he's trying to kind of illustrate is that in AB blood, you get a co-expression or a co-dominance where both are shown. You get A and B genotype. So you produce both A and B antigens. And if you took the lab, you see this in the lab because we looked at the genotypes of the fish and the co-expression of genes in the fish and their trans genes. So the blue fish and the red fish, did they make a purple fish? If they did, they were saying that it's co-expressed, that both red and blue genotype or alleles are in the genotype, and that's causing it to look a certain way, look purple instead of red and blue, because both red and blue are being expressed. That's different from incomplete dominance, where you get a phenotypical scale between the two. Pretty sure that's correct. I'll revisit this. let's move on. Sickle cell anemia is another example of co-expression because you get both types of hemoglobin. You get hemoglobin A and hemoglobin sickle. So if you have both hemoglobin A's, you have normal. If you have hemoglobin A and hemoglobin S, you have some sickle, but some normal. So you can produce enough normal cells that can accommodate for anemia, which is lack of oxygen in the blood. If you have hemoglobin S and hemoglobin S, you have severe anemia. So your cells are not transporting enough oxygen to your muscle tissue and all the other places that need oxygen, and all of your cells are sickle. This can be considered codominance as well as overdominance. Codominance would be seen here where you get some sickle and some normal. I think that's maybe a better example of codominance than the blood types. I think the blood types maybe I confused myself and I probably confused you too, so I'm sorry. But I think if you look at that and try to understand the way that the blood is donated, you'll get it. So HGBA dominant with respect to anemia. So if you have HGBA, you won't have anemia. It's dominant in terms of having anemia or not having anemia. But it's codominant with respect to the shape. So they're saying that you can get codominance. You'll get both cell shapes, but usually with a heterozygote sickle cell person, you're getting enough normal cells to survive. You probably still have some times where you don't get enough oxygen or you can become anemic, but overall, the normal cells outproduce the sickled cells. Now, overdominance in sickle cell anemia happens when you get another thing affecting it. So an external force affecting the way that the gene is inherited. In this case, malaria it's been known to be rampant in certain parts of Africa, and those same parts have shown increased sickle cell anemia. So what that's saying is when heterozygotes exhibit increased fitness relative to homozygotes, so that's saying that the heterozygotes here, which are displaying that, why isn't this working? That HGBR and and HGBS, this, which would be technically sickle cell, you get sickle cell and normal cells, has a better odd of surviving than the homozygotes, which have, and this is normal homozygotes, which have normal cell. The whole population has normal cells. What he's sort of trying to say here without saying it is that the people who have the recessive, or let me not say recessive, but let's say they have HGBS, these people don't live long. Which they can, and we talk about that later in this slide, but for right now, just consider that they're not going to have a high chance of living without some help. But in these places where heterozygote sickle cell anemia was being observed in the population, they also observed that those people were resistant to malaria. So mosquitoes could bite them and mosquitoes carrying malaria would bite them and they wouldn't get malaria. So that ended up, malaria is really bad. It can kill lots of people. And so these people who had these heterozygote sickle cell anemia traits, they were immune to malaria, and so they were surviving at higher rates than people with all normal cells. And so that codominance of the sickle cell and normal cells was actually increasing fitness in those areas. And so here is the difference between dominance, incomplete, and codominance. Maybe read over that. I'm not going to go over that. I think that's pretty self-explanatory. But here you can see these charts where you have complete dominance. I think this is in I think this is in just anything, where complete dominance, if you get to this halfway point where it's heterozygote, it's phenotypically dominant, even though it's recessive, or hetero, sorry. Incomplete dominance, you get this range. So you don't, just because you're here doesn't mean you just get dominant alleles or the dominant phenotype. You could get any variation of these. Polygene is going to be talked about later. I don't even really know what that chart is trying to tell me, but they're saying, I guess, that part of the population with gets a phenotypic. Yeah, I'm not really sure about that. I'm not even going to try. Overdominance, this is the thing that's talking about the sickle cell anemia and the malaria mosquitoes. This is showing you that the phenotypic value is actually higher in the population than either either homozygote, recessive or dominant. So there's more of these in the population than the others. And that's what I say down here, more frequently seen in population, and that's caused by something in the environment. If the malaria mosquitoes are killing people with this, or if sickle cell is killing these people and mosquitoes are killing these people, then these people are going to live because they're immune to it. And they have enough regular cells to keep them out of anemia. Whoops. Okay. So when we look at Kuhnel's odd yellow mice, he discovered that Mendel's laws applied to animals. Okay, that's sort of something we knew. That 3 genes were responsible for producing 2 enzymes that affect a single phenotype. So 3 genes responsible. So I think this is sort of dealing with. I don't love this point. I'm not quite sure I understand what that point's trying to convey, but he described the first multiple allelism, which is saying that there's more than one allele controlling a single gene. Eye color is a good example of this because you've got brown eyes, blue eyes, and green eyes. So there's 3 possibilities, but you can only pull 2 alleles. Everyone can only have 2 alleles for that gene. So even though there's a possibility of several things in the population, you can only hold 2 of those. So multiple alleles in one gene can have many mutant alleles. These alleles can be in different doses. So many alleles exist in the population, but people can still only carry 2. So I saw an example somewhere that was about, like if you had a deck of cards and each person in like there was only 3 types of cards. So say there was 1, 2, and 3. You can only pick 2. So I might have 1 and 3. And you might have 1 and 2, or someone else might have 2 and 3. So even though these are the possible alleles that you can pick from, you can only have 2 of them. And they can be expressed in many different ways. They could be codominant, they could be recessive. One could be complete dominance, they could be in doses. He's just trying to say that there's multiple allelisms at a single locus. So there's more than just black and white. There's black, white, gray, pink, purple, and it just depends on what you inherited that determines your genotype. So if there's a pool of a bunch of things, you pick 2. That's all you got. So I think, and then they talk about he discovered lethal null mutations. I think that's probably more of the important part of this, just like this guy's discovery, because that's what we talk about more in this PowerPoint. So a null mutation is going to completely disable a gene. And then if that gene codes for a protein or enzyme, then that enzyme or protein is completely non-functional, or it's not made at all. If that protein was something that you needed to live and it does not make that protein, then you are dead because it did not make the protein. So an example of that would be if a gene was controlling for heart muscles, or a pituitary gland. I think there's a fish that has a gene that codes for several things. And one of the things it codes for is the pituitary gland. It also codes for this this fin on the back of its back. Its back fin. I don't know what they call those. I know it has a name, but I can't think of it right now. If the gene turns off for this, it's dead. If the gene turns off for this, it's okay. It's okay. As long as it still codes for the pituitary gland. So that's sort of what a null mutation is describing. It's describing something that completely disables the gene and makes it completely non-functional. So we see this in mice. If you're pure breeding a white mouse and a gray mouse, and the outcome is, in the F1 generation, that they're all gray, maybe you get a phenotypic ratio in the F2 generation that is 3-to-1, gray to white, then you can say that the gray mouse is dominant to the white mouse, or the gray allele has complete dominance over the white allele. This must be complete dominance. And this follows Mendelian's law. However, and so you see that here, they use G as the dominant and W as the recessive. But so this would be white, this would be gray, gray, and gray. And all the mice live. Happy ending. If, however, you take a yellow mouse and another yellow mouse and you get a 3-to-1 ratio, that would still suggest that the yellow allele is dominant to the gray allele, because you get this is a Mendelian ratio. That's what they call those. But this guy, Clouteux, or however you say his name, Coulot, he discovered that, so this is just saying exactly what I said above, he discovered that instead of getting that 3-to-1 ratio, he gets this 2-to-1 ratio in the F1 generation. Because one of the mice dies. The only reason they get a 2-to-1 ratio is because every 1 out of 4 mice dies. That's yellow. It's a yellow mouse that dies. So what he discovered was that these yellow mice that have 2 dominant yellow alleles these dominant alleles are coding for something else that's making a null mutation. And that mutation is causing a lethal reaction, because whatever that is coding for or not coding for is essential for the mouse to live. So if the mouse gets 1 yellow dominant allele and a gray recessive allele, it lives. It's fine. The dominant allele, it's not affected by double dominance. So homozygous yellow dominant mouse is killed because yellow alleles also affect a gene that is essential for survival. So in a sense, and he sort of said this in class, that double dominant yellow allele is acting like a dominant allele for color, but it's acting as a recessive allele for something else that's essentially important. So it's a mutation that's recessive that turns off a coding area that's essential for life. So they concluded that the yellow alleles are lethal when they're homozygous. Mendelian inheritance still fits with yellow inheritance patterns. So you still get a 3-to-1 ratio of yellow to gray mice, but one of those yellow mice is dead. And since we know that you get these relationships of dominant, 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 recessive, dominant, recessive, recessive, recessive. They're saying that, well, if it's yellow and it's yellow's dominant, then yellow must also be acting as a recessive trait for something else that's killing the mouse. It didn't produce something that was necessary for the mouse to live. So allele capable of causing death, that's a pheno equals phenotype. I don't know what that means. Sometimes his slides just do not make sense. Demonstrates the importance of the gene's function, pattern of lethality, provides clues to function. Death phenotype may also indicate gene function. Leads to non-Mendelian surviving ratios. So he's talking here about that 2-to-1. Because one of the mice dies. That would be the non-Mendelian for the surviving progeny. So death phenotype may also indicate gene function. That's saying, well, if we know it's a yellow mouse that's dying, then maybe yellow is coding for something else. So here you get genotypic segregation. All right, gametes. So this is just sort of explaining all of that. The parental generation, you've got 2 yellows. You know they must be heterozygotes because they're both alive. And then they produce these gametes, which are, heterozygote. You would expect from a heterozygote. And then in the F1 generation, if you get that YY double dominant, you have a dead mouse. It's yellow. This is a yellow dead mouse, but it's also dead. And if you have 1/2 that are yellow and alive, then you must know that something's affecting the yellow mice. And it must be a dominant thing, because if yellow's dominant and these are non-dominant, then you're getting a can be dominant for one thing and recessive for another. So exactly. So you're getting a dominant in color, but recessive in lethality. I hope that makes sense. Y is dominant to Y for coat color, but YY is lethal. And this is called recessive lethals. Right. So Manx cats are another example of this. There's a gene that codes for their tail or absence of it, and it also affects their spine. So if you get a heterozygote cat, it lives. But if you get a homozygote cat, homozygous dominant cat, it dies. And it doesn't have a tail, but it also doesn't have something essential in its spine. And so that's what they're saying here. A single dose of the ML spinal affects the spine, but develops the lack of a tail. But a double dose of that causes malformation and then death. I have 2 Manx cats, so mine both don't have tails. They can produce tailed cats. That would be a recessive Manx cat for that gene. But it's recessive if it has a tail. It's recessive for the tail gene, for the why won't this erase? It's recessive, so tailed cat equals recessive for the ML gene. So it's just M. Oh my goodness, MM. But a heterozygote would be non-tailed, and a double dose or a homozygote that is MLML would have no tail, and it would be dead. And so homo or hetero equals MLM, no tail, alive. Okay, pleiotropy is when one gene controls multiple phenotypes. So we see this in both the yellow mice and the tailless Manx cats, but most recessive lethals are not polytrophic. So most lethal recessives just control one gene is what is what he's trying to say in this point here. But polytropic means that one gene affects two types of things. So it affects more than one. I guess it's, one gene. Mutations produce multiple effects. Oh, one allele, sorry. One allele affects more than one gene. That's what pleiotropic means. And most of the recessive lethals are not pleiotropic, which means they don't usually affect phenotype. So if you had a non-pleiotropic lethal recessive, it would be homozygote and dead, and it would not look any different than the rest of the progeny. But yellow coat and tailless Manx cats are examples of pleiotropy and lethal recessive mutants. I hope that makes sense. Okay. The environment can have an effect on your phenotype. This can be seen in Siamese cats. And lethality can be mediated by exogenous conditions, which means things outside of the body. So temperature, medications, things like that. So they can treat cystic fibrosis and they can treat sickle cell anemia with medications, even though those things are technically can be lethal. So if you had a sickle cell person who had the HGBS, they produce all sickle cell. I'm just going to put it that way. But if you give these people medication, they can survive. And the same is true in, apparently, modified agricultural crops. Who knew? Sublethality, consistent skew of phenotypic proportions, only some lethal allele homozygotes are affected. So sometimes homozygotes don't die in these progenies, even though they're homozygotes that are affected by the lethal allele. And that's usually due to something like meds, which is exactly what I was talking about here with the hemoglobin sickle cell anemia. These people can be provided medication and they live, so you actually get, what they do in sickle cell anemia is they actually provide the mother with more fetal hemoglobin, which carries oxygen better than other blood cells. And so that actually helps them. They can also get blood transplants, and there's medications that help with that as well. So these environmental dependent phenotypes, we see this in rabbits. We also see it in Siamese cats. He's all over the place here, but I'm going to try to keep pushing. Conditional mutants. Okay, so that's called conditional mutants, when things from outside change the environment, or the environment affects phenotype. So these organisms or individuals have the same phenotype in both conditions. So this fly has the same phenotype as in 18 degrees than it does in 28 degrees. It didn't change its phenotype. It just changed how much of that enzyme or product from the gene is being exhibited. So this is for individuals. This individual can change its phenotype from temperature to temperature. We see it in Siamese cats. Wow, there's nothing else on that. Great. Siamese cats have, let's see if I can draw a cat for you. Whee, there he is. And a little tail. Okay, Siamese cats have darker ears, darker tail, and darker paws than the rest of their body usually, and they have a dark nose. That's because these areas are extremities that are colder than their bodies. In winter, in winter, Siamese cats tend to get a little bit of a darker coat on their body because their fur is exposed to colder temperatures. And then in summer, they tend to turn back to more of that white coloring. This is an example of these conditional mutants. They're most often temperature sensitive, so just like Siamese cats. Permissive temp, wild type. Okay, so permissive temp means that well, I don't know if that means it's a certain temperature, but you'll get a wild type phenotype. So in Siamese cats, because I know cats better than I know flies, the permissive temp would be warmer. For Siamese cats, this is for Siamese cats. Because they're showing a more white coat. In the restrictive temperature, which would be colder in Siamese cats, they show a darker coat. Darker coat. So this just has to do about how the enzyme or the gene, or the enzyme that the gene is coding for, reacts in certain temperatures. That's called conditional mutants. You see it in these rabbits too. Just like the Siamese cats, their ears, nose, and tail and feet are all exposed, and you get a change in phenotype. These rabbits, apparently, when they get into temperatures above 30 degrees, their ears, nose, and tail and paws look wild type. So yeah, these are the same individuals though. Okay, that is chapter 10. I hope that made sense. If it didn't, you can let me know.